Hello my dear students, this is Dr. 𝗵𝗺𝗴𝗿𝗺, your biochemistry faculty. Well, in this video we are going to discuss a very interesting and an important topic, replication of DNA. So let's start with the discussion. DNA replication. Now see, we all are familiar with the DNA. What is DNA? Deoxyribonucleic acid. We know the structure of DNA. We know the composition of DNA. Now question is, what is a replication? In which phase of cell cycle it takes place? What is the advantage of this? And how this process is there in the body? So first of all, if we talk about this, DNA replication, always remember it is semi-conservative in nature. Very important. It is semi-conservative. What do we mean by semi-conservative? That half of the parental strand will be conserved in successive generations. Half of the parental strand will be conserved in the successive generations. Then now the question is, in which phase this DNA replication occurs? So this occurs in the S phase of cell cycle. Synthesis or replication occurs in the S phase, because S phase means it's a synthetic phase where synthesis will occur. Then coming over to this, first of all we should know how the flow of information is there in the cell. So if we talk about this, DNA forms RNA and this RNA translates the information in the form of proteins. DNA is forming DNA. This process we refer to as replication. So formation of daughter DNA from the parent DNA is replication, right? So I will write replication. This is what we are going to study here. And when DNA forms RNA, that process is transcription. And this RNA translates the information by a process known as translation. And this RNA, it forms DNA by reverse transcription. Very important. So now we are going to study this process. If we talk about DNA, we know that the DNA is different in prokaryotes and different in eukaryotes. So obviously replication has to be different in prokaryotes and eukaryotes. So if we talk about the prokaryotic DNA, right, we know that DNA is lying as it is in the cytoplasm. Why? Because prokaryote means primitive type of DNA is there. That means that DNA is not enclosed in a proper nuclear membrane or nucleoplasm. Everything is lacking there. And the type of DNA which is existing in the prokaryotes, it is circular. So I can say prokaryotic DNA is circular, known as a nucleoid. Then coming over to eukaryotic DNA. Eukaryotic DNA is a complex type of DNA, or it is a true nucleus. It is having a true nucleus in which DNA is present, which is linear in structure. And we know it is double-stranded also. Okay, so what is the process of replication in prokaryotes? What is the process of replication in eukaryotes? Because eukaryotes are highly complex organisms. So obviously it will be complex here. So the enzymes also will be different, everything will be different here. So we are going to study the prokaryotic DNA replication in detail and what are the parallel enzymes which are present in eukaryotes. Now coming over to this, we know that DNA is double-stranded. So first of all, we have to check what is the origin of replication, because DNA is lying as it is. So selection of origin of replication is very, very important, because it is not possible to you can say replicate all the base pairs at one time. So we have to select the origin of replication, and which is known as 𝗲𝗏𝗃, or the origin of replication. So obviously prokaryotes and eukaryotes, they will have different origins. So prokaryotes will have a single origin of replication, single 𝗲𝗏, and whereas eukaryotes will have multiple origin of replication. Very important, right? So now the question is, when the origin of replication is selected, then different enzymes will come, because we have to unwind the helix. Okay, when we are unwinding the helix, then other enzymes are going to act on it so that the daughter DNA will be produced. So this process is not that easy process, because whenever it's like a rubber band, so whenever we try to pull the DNA strands apart like this, somewhere it will be overwinding, somewhere it will be underwinding. So whatever stress and strain is produced, we have to release that also. So it's like a very complex procedure which requires so many enzymes to work with. So what are the list of the enzymes when the origin of replication is selected? They are the enzymes required for replication. Right. So if we talk about the enzymes, the most important enzyme here is helicases. Now what is the function of this helicases? First enzyme, it is unwinding the helix. So the first function is unwinding of helix. Right. Then once the helix is unwound, then some kind of supercoiling is introduced in it. So it generates negative supercoiling or supercoils, right? So helicases is just unwinding the helix. Then second is a very, very important enzyme, we call it as a topoisomerases. What is the function of this topoisomerases? Because what are the topological properties of DNA? We all know that DNA is having different type of bonds and it is intertwined like a rubber band. So we cannot just pull DNA like this. Whenever we are trying to pull the DNA strand apart, so what happens is so much of stress and strain is caused. So the purpose of this enzyme is to release that stress and strain, because whenever DNA has to undergo a process of replication, it should be stress free. So for that, the enzyme is topoisomerases, and we call it as a stress relieving enzyme. Very important. Right. So whatever negative supercoil is generated, it will try to convert it into positive one. So it helps in generation of or generates positive supercoils and reduces the strain. And this topoisomerases is of two types. One is topoisomerases 1 obviously, and the other is topoisomerases 2. And what are the different functions? Topoisomerases 1 cut one strand, 2 cut both strands. No ATP required. And in topoisomerases 2, ATP is required because it will cut and rejoin. It is going to cut both the strands and it is going to rejoin them. So for that purpose, energy is required. Then third is gyrases. Now what is the function of this gyrases? This gyrases is present in the prokaryotes. So it's like present in prokaryotes and serving the purpose of both. So both means topoisomerases and helicases. Then fourth important enzyme is, now this is a very important enzyme, in fact we call it as a primase, right? So this primase is already having say 5 to 50 nucleotides, okay, and it's an RNA primer which is dependent on DNA. So it's like an RNA primer dependent on DNA. So we can also call it as DNA dependent RNA polymerase. And this primer will just initiate the function. Once the function is over, it is removed also. So I can say having 5 to 50 nucleotides, RNA primer dependent on DNA just initiates the process. Initiates the process and then it is removed. Process then removed. Then we come over to the last one, which is known as SSB, which stands for single strand binding proteins, which always stabilizes the single strand of DNA, because DNA is going to unwind like this and it is going to form a replication fork. So single strand binding protein will be there, which will prevent that reunion of that, because we have to synthesize the daughter DNA. So it is single strand binding proteins, right? And what is their function? Their function is to prevent the joining of the strands, to prevent the joining of strands. And then we come over to the last most important maximum fidelity enzyme, which is DNA polymerase. Right, now this DNA polymerase, which is the main enzyme here, as it is catalyzing the polymerization, and it has to be DNA dependent DNA polymerase because we are synthesizing DNA. So we can say it is simply DNA dependent DNA polymerase. Very important. Okay. So what is its function? It is simply accelerating the formation of daughter DNA, and this DNA polymerase is of different types in prokaryotes and eukaryotes. So if we define it, we can simply say that in prokaryotes and in eukaryotes, right? In prokaryotes, DNA polymerase is of 3 types. So we can say DNA poly 1, DNA poly 2, polymerase 2, and DNA polymerase 3. So if we talk about polymerase 1, the most important function is primer removal. If we talk about 2, mainly it is in repair, and third is polymerization, or you can say formation of the daughter polymers, polymerization. And if we talk about the eukaryotes mein it is of 5 main types: alpha, beta, gamma, delta and epsilon. And the comparative, I'll just show you the picture also. What is the comparison between the two forms? This is very, very important, which one has to learn. This is a very important table in fact. Now see, if we talk about the major DNA polymerases. Prokaryotes, just now I told you, mainly it is poly 1, 2 and 3. Now 2 more have been discovered. And here it is alpha, beta, gamma, delta, epsilon. Now the main replicating enzyme, or which has maximum processability, it is polymerase 3, as you can see here, and here it is for lagging and leading strand. For this also we can say gamma. So if I write it like this, lagging mane G, so G for gamma. So I can say that lagging strand synthesis. And if we talk about GG, so you can say gamma is for lagging strand. Abhi right now, after this I will tell you what is a lagging strand, what is a leading strand. And if we talk about leading, so it is LEAD, leading strand. So E, epsilon. So this is just a mnemonic, just to remember how we can remember these, because these are very, very important from examination point of view also. Then if we talk about primase activity, primase is what? Primase is that RNA primer which is generated by DNA polymerase, DNA dependent RNA polymerase, which is already having 5 to 50 nucleotides. Here we can say the DNase enzyme is there, not in poly 1, 2, 3. And here we can say poly alpha has this primase activity. Primase means removal of the primer. Then if we talk about proofreading, proofreading is a very important, I would say, mechanism. What happens is, just suppose if we are writing an exam, whenever we have to submit an answer sheet, we will always go through it once or twice. That is proofreading. Because DNA has to undergo proofreading, otherwise if DNA goes wrong, so automatically the entire central dogma of information will be wrong. So proofreading 3' to 5' exonuclease. So it is present in poly 3 because it is the main enzyme for processability, and poly 1. And here if we talk about, yes, poly, this thing has, but poly alpha does not have a proofreading activity. Then removal of RNA, poly 1, as I told you here, poly 1 is required for primer removal. And here RNase H and FEN, they are the other ones which are required for the polymer, this primer removal. Then this is mitochondrial, and if we talk about DNA repair, poly 1, 2, 4, 5, and here poly beta is there. And then this replication speed, it is fast, it is slower, because DNA polymerase 3 is the enzyme which is having maximum processivity. So this is a comparative statement, this is a comparative kind of thing between the prokaryotic and eukaryotic DNA polymerases. And always remember, DNA polymerases is the most important fidelity enzyme of replication. Now coming over to this, we have selected the origin also of replication. So generally we see that origin of replication is having TTAAGG kind of sequences. Okay. So now the question is that once the origin has been decided, once topoisomerases has acted upon it, once the strand is open, then the process of replication will start and it is going to form a replication fork. Now once the replication fork is there, another very important point which you have to remember is new strand, whether it is DNA, whether it is RNA, is always synthesized in 5' to 3' direction. This is very important. New strand will always be synthesized in this direction, be it DNA, be it RNA. Now coming over to this, how this process takes place. And this is a type of replication fork, like this. So what happens is, as we have studied in the structure of DNA also, that DNA is having anti-parallel strands. That means if one is running in the direction 5' to 3', The other will run in the direction 3'' to 5'. Okay. Then RNA primer is already generated, right? And here single strand binding proteins are there just to stabilize the helix so that it cannot reunite again. Now the question is, I told you that new strand will always be synthesized in the direction 5' to 3'. So here, just to maintain the complementarity and the anti-parallel nature, here though we can say the strand can be synthesized like this, 5' to 3', because this is the growth of the replication fork. Replication fork, right? So one strand can be easily synthesized just to maintain the complementarity and the anti-parallel nature of DNA. Now the question is, एक तो DNA replication is semi-conservative, okay. दूसरा, when the other strand has to be made, it has to move to the opposite of the replication fork growth, because it has to maintain the anti-parallel nature, which is very, very important. So just to maintain the anti-parallel nature, it is going to require so many primers. But you can say that 5' to 3', 5' to 3', small fragments will be generated like this. And all will be in the direction 5' to 3'. That means they are maintaining the anti-parallel nature. All will be in this direction, right? Now since they are moving against the growth of replication fork, they are broken into fragments, and these fragments were discovered by Sir Okazaki. That is why these fragments are known as Okazaki fragments. Very important. And since this process lags behind the process, because one will be immediately synthesized, so this is known as the leading strand, and this lags behind, so it is known as the lagging strand, right? So basically what happens is that since new strand is always synthesized in the direction 5' to 3', so I can say that to maintain the anti-parallel nature of DNA and 5' to 3' condition, it moves against the growth. It moves against the growth of replication fork. Very, very important. Replication. And that is why it is broken into fragments. So fragments are Okazaki fragments. Zaki fragments, and the strand is the lagging strand. Right? And the other strand will be the leading strand. Very important. Right. So you can say that this is how the process of a replication will occur, and slowly the primers are removed later. And always remember that there are only two processes in our body which cannot initiate without a primer. One is glycogenesis, where the primer used is glycogenin, and here the primer used is RNA primer which is dependent on DNA. And once the primer is removed, then the nix or the seal, nick is sealed by DNA ligase. Right. So I can say that the two fragments, once they are synthesized, the two strands are synthesized, then the primers are removed, and whatever nick is there, it is sealed by this DNA ligase enzyme, which is also known as the glue of DNA. Also known as glue of DNA. And if we talk about the first reaction, what is happening here is, say this is the replication fork like this, and say this is the primer. So what happens is, just to maintain the complementarity, say adenine, thymine, guanine or cytosine is there, TTP will come. So I can say that T, A, C, G, these bases will come and it will liberate PPI. So basically, what type of reaction this is? This is a nucleophilic reaction. Right, where pyrophosphate is released, giving energy to this reaction to occur. So always keep this thing in mind, that this is how the replication takes place. Okay, let's take a quick recap. First of all, you should know what are the enzymes required for replication. So I told you the most important enzymes is, first of all, we have to unwind the helix. Unwinding of helix is done by helicases. Whatever stress and strain is produced in that, because DNA is completely intertwined, so many supercoilings will be there. It will be negative, it will be positive. Topoisomerases will come into function, which is going to release these supercoils. How? By either cutting one strand, so it is topoisomerases 1, or by cutting two strands, and then whatever stress is there, give out that stress, and again it is sealed up. Single strand binding proteins will come and they will just tell DNA not to anneal again, because what we have to do is we have to form the daughter strands. Prokaryotes are lacking helicases and topoisomerases, though the function is taken over by gyrases there. And then comes the main one, which is DNA polymerases. Now DNA polymerase is 1, 2, 3 in prokaryotes, alpha, beta, gamma, delta, epsilon in eukaryotes. How you have to differentiate? Always remember, DNA polymerase 1 always helps in removal of the primers. 2 is responsible for the repair function. Repair function I will discuss here. And the third one is the basically enzyme which is giving maximum processivity to DNA. On the other hand, alpha, beta, gamma, delta, epsilon mein, the most important point you can remember is, if we talk about eukaryotic, just to remember it, eukaryotes mein, alpha primase removal, right? Beta is for mitochondrial DNA, because mitochondrial DNA is altogether different. Alpha, beta, gamma. Gamma is for lagging strand synthesis. Very important. And epsilon is for leading strand synthesis. Now, to maintain the complementarity, to maintain the anti-parallel nature of DNA, what we do is, then proofreading is done. And one strand is running in the direction 5' to 3', which is along the growth of the replication fork. The second strand, to maintain the anti-parallel nature of DNA, or since new strand is always produced in the direction 5' to 3', it moves against the growth of the replication fork. So obviously different primers are required. The primers are required in small fragments may it is produced. But yes, the mandate is maintained. Means 5' to 3'. Later on, these nicks are filled by DNA ligase and we get the entire strand. But since this process, the strand lags behind the first strand, which was the leading strand, this is known as the lagging strand. and these fragments are known as Okazaki fragments, right? Now after this, then what happens is there is a type of proofreading which is a very, very important activity. So proofreading or editing is done. Now what is this proofreading or editing? There is this DNA polymerase moves along the DNA. So wherever it finds the mismatched base pair, it will just fix itself there and it will try to remove that. So different type of activities are there. 5' to 3' exonuclease activity, right? 3' to 5' exonuclease activity and 5' to 3' polymerase activity. Right. So what happens here is this is the structure of DNA polymerase 1. So what happens is that this is N and this is C-terminal. I can say that it is having 3 fragments. One is 5' to 3' exonuclease, right? This is 3' to 5' exonuclease and this is 5' to 3' polymerase. This is the structure of DNA polymerase 3. Okay, so now what happens is basically 5', 3', it's but obvious from the direction it is going to cut. So there is one bacteria, Bacillus subtilis. In there that subtilicin protein is there which is going to cleave here. Okay, so this DNA polymerase 3 will be cleaved into 3 sides. This is the shorter fragment, this is the larger fragment. So when this shorter means if we take this 5', 3' exonuclease activity out, this is known as Klinov fragment. Okay, so Klinov fragment is the one from which 5' to 3' exonuclease activity is removed. Right. So this Klinov fragment is basically having what? This type of structure, 3', 5 exonuclease and 5' to 3' polymerase activity. Now the question is, what is its use? So this Klinov fragment is required. There are so many uses of this Klinov fragment. Yahan pe to it is of no use, but yes, once it is cut, this Klinov fragment is required for conversion of sticky ends to blunt ends. This is suppose if we have a DNA strand like this. Okay. So what will happen is that this part will be removed. So what happens here is, we can say Klinov fragment. What it will do is, it will cut this part and it will join it here like this. This fragments will be removed like this. And what we get is 5' to 3', and this is formation of blunt ends. And this is sticky ends. So this is one function of this. Then DNA polymerase 1 is also known as Kornberg's enzyme. So if we talk about the properties of DNA polymerase 1, it is known as Kornberg enzyme because it was discovered by that scientist, Kornberg. Okay, So whether it is Glider fragment or you can say like this, DNA polymerase 2 is helping in repair function. So we have to just check the mismatched base pairing. So if any mismatched base pairing is there, immediately DNA is repaired. Okay. So DNA repair we are going to again discuss in a separate video. But here in this video you should now know what is the use of this DNA replication, the most important one. What are the enzymes required for DNA replication? What are the steps? So if I talk about the steps, we can just focus on the enzymes. These are the 5 main enzymes which are required. Then the Fidelity enzyme is DNA polymerase. And the most important process, you can say 2 main important, I would say points which distinguishes replication and transcription is here. Replication cannot start without a primer. Proofreading is the most important one. In transcription, you will see no primer is required, no proofreading is there. No doubt modification is there, but proofreading is not required. So this is what is the entire DNA replication. So I hope you have understood this. That's all in this replication of DNA, which is semi-conservative in nature. Thank you so much.